Right. Okay. Right. So, hello everyone. Thank you for joining us for this live Q&A question and answer session with Dr. Brooke Johnson today. And I would like to briefly introduce Brooke before I actually tell you about the format that we're going to follow for today's session. So Brooke had completed his geology degree at University of London and then moved to Oxford, to University of Oxford, to do his doctoral research on understanding environmental changes that happened 1.4 billion years ago during the Mesoproterozoic era. That is quite long. We need to find a lay terminology for that. So he's passionate about public engagement and he has been involved in several events, and he's particularly interested in working with pupils who might not consider further education or usually have access to earth sciences or higher education. So before we start our discussion, I would like to remind everyone about our structure or introduce you if you're new to Live with Scientists Q&A sessions. So the structure of the session will start with a discussion where I will be asking Brooke some questions for about 40 minutes. And then we will have a 10-minute break to give our audience an opportunity to submit their questions through the comment log on the right-hand side of the YouTube page. So after the break, we will resume and we'll have a further Q&A session for another 20 minutes, during which I will be asking your questions on your behalf. So Brooke, thank you very much for joining us today. It's really nice to be able to host you during this session. And yeah, if you're ready, I would like to start with our questions though.
Yeah, let's do this.
Yeah. So I have got a list of technical questions though. So obviously you do separate, you have got several geological eras. So can you please tell us what's the importance of the Mesoproterozoic era relative to others and why you decided to focus on that? And if we can find a shorter or a lay terminology, I would really appreciate it.
Yeah, we'll call it the Middle Age.
Fantastic, thank you.
Because it's Earth's Middle Age. So
right.
if you imagine that, like, you hold your arm out,
yeah,
one of your arms out at arm's length, and if you imagine that Earth formed 4.5 billion years ago, and that's the middle of your chin,
yeah,
all of the animals and fossils and things that we can see, they only appear round about the bottom of your wrist. So the Earth's Middle Age is most of your forearm.
okay.
From like your elbow to your forearm. The rocks I study, this also might help you understand how old they are. They're about 5 times older than the oldest dinosaur. So the Earth back then, 1.5 billion years ago, all of the continents were joined together into a continent called Nuna. There was one giant ocean. A lot of the land surface was flooded by shallow seas. There was nothing alive on land except maybe some bacteria. And the ocean was completely dominated by bacteria. There was, as far as we know, very little oxygen in the atmosphere. So if you went back in time there, you'd be stood on this desolate landscape. There's no ozone layer, so you'd need lots of sun cream. You'd need an oxygen mask because there's also a lot of carbon dioxide. The whole planet would have been really, really warm as well. There was no ice at the poles. And if you'd have gone to Northern Australia, then the area that I look at, which is now the desert, was a shallow sea, nice warm shallow sea, and it would have probably been green with piles and piles of bacteria that were living there because there's no animals to eat them. So you'd have been stood on this desolate landscape with this green sea in front of you, and the moon would have been bigger, and volcanoes and everything you'd imagine that the primordial Earth, the ancient Earth would have looked like. But the reason this period of time is so special is because this is when complex life first appeared. So all life that we know of falls into 2 groups. You've got bacteria, which are just basically like a little capsule or a little pill full of soup, and they're alive. They can wiggle around, they can reproduce by splitting up, but they're not very complex. They can join together to do bigger tasks and things. All other kind of life is called a eukaryote, And that's everything from an amoeba, a single-celled amoeba or an algae, all the way through to us, to the biggest whales and trees, funguses, everything that you can think of that isn't a bacteria. And our cells are quite complex. They have little engines inside them and little organs that do specialized tasks. And we don't just reproduce by splitting, although we can do that. We can also have like sexual reproduction and swap genetic material. And the leap between a simple bacteria that's basically just like a capsule or a can full of soup and this really complex cell structure is like it's like a mind-blowing thing. It's a bigger gap than going from Stone Age technology to spacecraft. It's such a huge gap. And the time I study is when that happened. And we don't know why it happened, but there's a bigger mystery, because when eukaryote cells appear, they then do nothing for 1 billion years. So they're basically just sat there kind of existing. They're not diversifying. They're not evolving. They're just kind of plodding along for 1 billion years, and we don't know why. So that's what that's kind of what I look into, and that's what was special about this time period.
Okay, all right. So we knew that then there was some form of life present at that time. You know, we definitely know that there were plants around, there was microorganisms around, but nothing complex, nothing multicellular is what you're saying, apart from plants.
So not even plants.
Okay.
No plants at all. Plants don't come along until much. I think animals actually turn up before plants.
Okay.
But plants as we think of them don't exist. So there are bacteria that photosynthesize around now that do the same kind of jobs as plants. If you think of like, they call it what animals do in an ecology is like a job, then there are things that do that job of photosynthesize. We call them cyanobacteria. And whenever you've seen like a pond or a lake or a stream, they're the kind of green, mushy stuff that you see. They make all of our oxygen. So they were around, but there's nothing modern around at all, nothing that you would recognize.
Okay, great. Thank you. So then I would like to ask then, you know, it is obviously so long ago. So can you tell us a bit more about, in layman's terms, what techniques do you use to study things that happened, you know, 1.4 billion years ago?
So I do a lot of drawing and colouring in. I literally draw the rocks and colour them in, which sounds facetious. It sounds like I'm being a bit silly, but I'm not. So when we get these rocks, they're underground, and we have a huge drill that pulls them out in a tube, and this tube of rock comes out in sections, but it can be like thousands of meters long. If you look on the website, that photograph of rock, those tubes of rock are ones we've pulled out, and you'll see they've got all different kinds of colors and textures. And those colors and textures tell us are records of the environment and of what life was doing at that time. So by recording those colors and textures, by drawing them and coloring them in and making what we call a graphic log, we can actually record how, and going along this tube of rock, we can record how the environment is changing. So that's the first thing we do. We just use our eyes, and we don't use any fancy technology or terminology. You're just kind of going, well, that bit's bigger than that bit's green, that bit's red, that's smooth and that's wiggly. And maybe so you'll use like a magnifying glass. Okay. So that's the first thing we do, and we can get a huge amount of information that way. Next thing we do is take those rocks and cut them in half and polish them and look at them with a binocular microscope. And that's basically just like a very fancy kind of magnifying glass. You have one on each eye, and so you can look at the rock really close up. And that gives us another layer of information. And then I'll take one of those chunks of rock and I'll slice it so it's about the size of a domino. And then I'll grind it down so it's about as thin as one of your hairs. And then I'll look at it under a different microscope that shines polarized light through it. And polarized is a fancy way of saying goes in a straight line. So light bounces all over the place and is all jazzy and very excitable. But if you have a polarizer, it's like a gate that forces all of the light to go in one direction. And when polarizing light passes through an object, it interacts with that object. It'll get bent and twisted a little bit because the structure of the crystals in the rock, the electromagnetic and the gravity of them, the forces will pull the light ever so slightly. and that lets us see change the. I'm getting too excited now. And what that does is it changes the color of the crystals in predictable ways that we've catalogued and we know. So we know which minerals we're looking at, and it lets us see textures we couldn't see otherwise, that let us know what was happening on the scale of microns. So a micron is like a thousandth of a millimeter. So if you think a grain of rice is maybe 2 millimeters long, so imagine the various tiny tip of a grain of rice. That's the size of things that we'll look at with this microscope. And we can see things like fossil bacteria, for example.
Okay. Fossil bacteria?
Yeah, fossil bacteria.
Okay. I mean, you know, if you have any of these videos, we would be interested to see this just before our Q&A session, if you have them available.
Yeah,
yes, that would
I
be
do.
quite...
I've got all of the pictures and videos.
Oh, that's great. So I think it would be nice, or if they're handy, we can maybe have a look at them now.
Sure, yeah, we can keep talking and I can dig them out. I did wonder if I should do any in advance, but
that's fine. That's fine. Or like I say, we can do it right before the Q&A session as well, whichever you prefer. So obviously you mentioned about fossils though. So can you tell us a bit more about how do they form?
Yeah, so a fossil is the remains of a living organism that's preserved in the rock record, and they can form in a lot of different ways, and they can preserve a lot of different things. So I think everyone's probably been on the beach at some point or by a lake, and they might have seen shells or bits of plants. But if those shells and bits of plants get covered by mud and successively buried by getting more mud piled on top of them, that mud will eventually turn into rock and it will preserve the organism as a fossil. And that can either happen by preserving the original material that it's made of. So things like shells, they're actually already made of minerals, they're made of stone, same as bones. And that they preserve quite nicely. Things like plants are actually quite resistant. So you don't preserve the original material, but you preserve all of their details in carbon. And sometimes it can be preserved so well that you can see individual cells. So there's a place in Scotland underneath a golf course, I believe, where there are some of the oldest plants in the world are preserved, and you can see their cells and how they were arranged. Yeah, so most of the time you just get hard parts, but there are very special occasions where you get soft parts as well. And then we can see things as things like eyes and guts and brains, and they get preserved by clay sticking to the organs and making like a template out of it. And some clays are antibacterial as well, but sometimes the bacteria help by precipitating, by basically pooping out minerals that preserve the organism. It's really complex. The study is called taphonomy, and it's like super fascinating. And sometimes you can even get fossils preserved by, say, volcanic eruptions like Pompeii.
Okay.
There are even ancient versions where South American rhinoceroses were preserved by a volcanic eruption like Pompeii.
Yeah.
But most of the time, you're going to get hard parts. But it turns out bacteria are quite resistant in natural environments. As long as you're not cleaning them with bleach and antibacterial, they'll survive quite well. And like I said, some bacteria basically excrete minerals, and so they'll accidentally fossilize themselves in certain circumstances. And that's what I find in my rocks. These little things that look like tiny, tiny, tiny wee polo mints, little hoops made of a mineral called apatite. And apatite is what your bones and teeth are made of. And so we know it's generally made by living organisms. And for a long time, I couldn't figure out what they were. They're about a quarter of the width of one of your hairs. So these things are tiny, tiny. Until I looked at them on an electron microscope, and then I was able to see them close up and see that they had structure, and then in a organic carbon core. And then I compared them with other people who'd found similar things, and we all had a different bit of the puzzle, and then went, oh, they're bacteria.
Okay, great. Thank you. So the other thing that I was wondering though, obviously you're seeing these structures in the rocks, but can you actually, as from a biologist's point of view, obviously I'm excited about, can you actually extract ancient animal DNA from fossilized, let's say, rocks from fossilized amber and do some analysis on it? Or is it too long that it won't preserve for such a long period?
Yeah, so DNA, it turns out, is really unstable. Things that have a lot of proteins in them are full of energy, and bacteria and other organisms will be straight on that at the point, like when an organism dies. So it tends not to survive that first level. It gets filtered out quite quickly. That's why, for example, in oil, what you tend to get are the resistant materials, the carbon-based materials from plants and bacteria and algae, because all of the proteins and fat from animals gets eaten really quickly. So amber is especially interesting because it gives you a perfectly preserved what's on the outside of the organism, but actually it desiccates and dehydrates the inside. So when you see those beautiful, and like in Jurassic Park, where people drill in and extract the blood, there's actually nothing inside there. They're empty, it's just dust. Amber's also not, it doesn't perfectly seal them. It's kind of freezable. So gas and liquids can get in and out over very long time scales. But you can get some very strange things preserved. Recently, and this is something I never thought I would say, but recently someone showed me an insect in amber that had farted while it died, and the fart bubble had been preserved.
No way.
Yes, yes, way.
Okay, interesting.
Because if it's not that, it's some other gas coming out the back end of the animal.
Okay.
However,
quite interesting.
Some people claim to have found fragments of DNA from dinosaurs that died in arid conditions and were mummified. So there's not enough DNA there to bring them back to life or analyze or understand in any kind of way. But there was enough there for them to say this was probably DNA at one point.
Okay.
The other thing that you've got to contend with is as rocks are buried, the temperature goes up and the pressure increases. So some rocks, if they're only shallow, they'll be about 150 degrees centigrade for thousands, if not millions of years, and then thousands of times the Earth's surface pressure. So a lot of soft and fragile biological material doesn't survive that kind of treatment. Yeah,
I assume so, yeah. Okay, so, in your field of research then, can you tell us, obviously you're seeing lots of different fossils and you're being able to actually do some research on them. How often do you think a scientist would have a chance to discover new species of extinct animals through fossils? Is it as common or as rare as we anticipate it to be? Or, can you give us some insights into that?
Well, it's not rare at all. We probably only really know a very small percentage of animals that are alive today, and the percent of extinct animals that we know is even tinier. And I mean, like less than 1% of all life that's ever existed on Earth. So it can be quite common to discover new species. A lot of the times people will do a bulk collection at a new site, and then they'll go in a museum collection, and then they'll forget to work on it, and people won't know about it until decades or even centuries later, when a grad student comes along and says, oh, what's in this drawer? So that's really common. The other thing is you have to define what a species is. And this is a bit of a contentious point, is that people don't really understand what a species is for living organisms. But it's even harder if all you've got is a bit of shell.
Yeah.
So where I grew up in North Yorkshire, there are lots of ammonites, and an ammonite is basically a squid with a fancy curly shell. And people like to divide them up into lots of different species because there are lots of different varieties. And some of them you can look at them and say, in the same way that you would look at a tiger and a lion and say, well, they're related, but they're obviously different. And some of them you would look at them and it'd be as different as looking at, say, like a tiger and a house cat, and you'd say, they're very different. But some of them, you would be like looking at like a two different types of tiger and going, well, how can I tell which one's which? And if all of you got a shell or a bone, it's really difficult. Think about how much variation there are between humans. How much of that variation is then lost when humans are just a skeleton?
Yeah.
Imagine, in my case, for example, I've had broken bones and I've got metal plates and I've got all kinds of like where I've fallen over and hurt myself. Would someone in the future look at those and go, oh, this is a separate species because look, it's got a metal jaw or what have you? Or would they say, actually, no, these are all the same species? And it's the same with fossil creatures like the ammonites. The splitters, which are people that like to divide things up, have divided them up into lots and lots of species. But the lumpers have said, these are all pretty much the same. They're probably one species. Some are probably male, some are female. Some had injuries, some are pensioners, and some are just young babies. That's why you've got all this variation in them.
Okay.
So it's quite a big argument. And sometimes people discover that a new species is basically like the what we call sexual dimorphism. It's either the female or the male version of one creature, or it's the juvenile or adult stage of one organism. And sometimes, in some famous cases, people discover that several separate species are different parts of the same organism. So there's a very famous ancient crustacean, ancient arthropod, so creature related to things like crabs and insects, that lived about 500 odd million years ago, called Anomalocaris, which means mystery shrimp, because people found its two front claws first, and they look like a shrimp missing its head. And then they found this thing called Patoia, which looked like a jellyfish. And then they found this other thing they thought was a sponge, and I can't remember the name of it. And so they classified them all as 3 new species. And then about 50 or so years later, someone found a complete one and realized the shrimps were the front claws. The sponge was actually the body of this big flapping creature that had lots of flaps to swim. And the jellyfish was its mouth parts. It had this circular mouth for grabbing and crushing.
Oh, I see.
So that sort of thing could happen. You can never be sure if you've actually found the whole animal or the whole organism, or just a little wee bit of it.
Yeah. Okay. Well, that's very interesting. Thank you very much. And it was very informative as well. So, so for a budding geologist then, where would you think would be a good place to start looking for interesting rocks or fossils?
That depends on where you live and what's available to you.
Okay.
The easiest place to maybe look at that is if you're in a big city, is the buildings around you.
Okay.
So just thinking of Oxford's got lots of buildings made out of lots of different types of building stone. So there's about 3 billion years worth of Earth history just in the building stones of Oxford. And Oxford, in terms of its building materials, is typical of most big cities. Well, I say big cities, most older cities in the UK. You've got all different types of rocks: sedimentary, metamorphic, igneous. You've got lots of fossils in those rocks as well to look at. And most people will walk by them every day. For example, Tesco's in Oxford Town Centre has a building stone called lavakite, and it's really common across the world as a decorative building stone. And lavakite contains these big crystals of a mineral called labradorite, which is also a gemstone. And people walk past it every day and never notice it. And occasionally I go and look at it and someone will ask me why you're staring at that wall. But when they notice it, the thing about labradorite is it has mother of pearl effect. It has rainbow colors on it when you tilt your head. And then all of a sudden people notice that there's this wall made out of all this shimmering rainbow colored material. So the best thing to do is have a look around of the buildings around you, if you've got that available. If you're in the UK, the British Geological Survey make all of their things free. Their maps, there's a digital app so you can find out what the rocks are like around you. And I know the USGS has a similar thing, but I don't think they have an app yet. Also the internet, there are lots of websites dedicated to different locations where you can go and check out and see what the geology is literally everywhere. If you just scratch the surface and once you start seeing it, you won't be able to unsee it. It'll be everywhere.
Lovely. So maybe you can drop these links on your website, on your space on our website, so anyone who's interested can actually have a look at it.
Yeah, so one of the things I did on my YouTube channel, sorry for the shameless plug, is that there's an episode where I walk around Oxford and show the different kinds of building stones. So you can see and they're all very common building stones in the UK, and even in the rest of Europe and North America. So so that'll give you an idea of the things to look for and the kinds of things you can see.
That's actually quite interesting, and we would be interested to include the link for your YouTube channel as well for those who are interested.
Oh, thank you. That'd be good. It's all in common language as well. It's designed for my dad to watch it, so
that's quite good. Yeah, we would be really interested to do that for our audience. So going back to the technicalities though, can you tell us how do, obviously you can't do very much of analysis on them as we know it. Let's say as a biologist, as I know it, you can't do very many analysis on them. But can you tell us at least how do you think those microorganisms are different from the ones that we see, the modern microorganisms are different from the ones that you see in distant past?
Well, they're not. They're not different at all. That's the thing. So you can do this thing called, and this is one of the big differences between bacteria and all other life, the complex life, is that bacteria kind of hit their peak and were like, yeah, this is it. They'd have evolved to fill all of their niches, so to speak, and they haven't really changed in the billions of years. So there's a technique called molecular biology. I say technique, a field of study, and people can kind of look at the timings of DNA. I don't really understand it because it seems like magic to me, but I have friends that do it and have tried to explain it to me. And you can kind of look at the rate of how the DNA changes to trace when different species changed and evolved. And people have done this to bacteria, and what they find is that bacterial DNA is really, really old. So, and because we share a lot of similarities with certain kinds of bacteria, we've incorporated in. They know it goes back a really long way. So the ancient bacteria I find in my rocks, pretty much the same as their equivalents in the modern day. So what I find are things like cyanobacteria. And when you look at cyanobacteria fossils from billions of years ago, they look pretty much the same. And then chemically and geochemically, with the bits that you can get out of the rock, you can tell that they're kind of doing the same jobs. And the other big one I get are sulfur bacteria, who make the phosphorus, who make the apatite. And that's exactly what they do. I've looked at sulfur bacteria that I've plucked out of a stream, and they're identical to the ones that I found in my rocks. But bacteria don't have very many shapes. But the important thing is that they're doing the same sort of jobs. They're producing apatite, they're processing sulfur and cycling it through the environment so it doesn't get stuck in one place too long.
Okay, great. And I know you mentioned about the methods that you actually use to get the material to work on, but can you compare the modern analytical techniques that you are using in your research to the ones, to the more traditional ones? Do you think you are in a better position than you were before in order to make some more discoveries?
So the analytical techniques they're just tools, basically. And if you think of instead of thinking of a scientific and analytical technique, think of a hammer. Like hammer's really good for hammering nails into stuff and pulling nails into stuff. It's good for splitting rocks open and finding fossils, but it's not very good for other jobs.
Yeah.
So what it comes down to is you can have a new fancy tool, but if you're not using it properly and you don't understand its limitations, then you might as well just have a brick. It's not going to do you much good. So what I found is, and what I've tried to do with my work, is take these traditional techniques that are starting to kind of get, say traditional, they're only like a couple of 100 years old at most. These techniques like looking at the rocks and using microscopes, and then tie them to the modern analytical techniques where we put things in a mass spectrometer and blast it with plasma and do all of these like things that to me still sound like science fiction, and tie the two together and use their strengths to kind of build a broader picture. Because if you were building a shed, you wouldn't just use a hammer. You would use other tools as well, and you'd use each tool for the right job. And they've definitely increased our ability to understand what's going on in the past. The new tools, that is. I tend to think of it as like viewing the past through rocks is like trying to look at a really complex picture, but all you've got is a keyhole. And then by having these new tools and using them in the right ways, that gives you another keyhole. And then by using them in conjunction, instead of just looking through one keyhole and saying, this is what the whole world was like for 1 billion years, you can go, okay, that bit's like that, that bit's like that, that bit's like that. And then you can put together a bigger picture of what was going on.
Okay. All right. Great. Thank you. So I have got a couple more questions if you're not too tired, though. I know you've been talking about for half an hour now. So
Doesn't seem that long.
yeah, I know, it just flew by, didn't it? So I'm also interested in learning about your personal experiences as well. So I would like to, can you tell us if there are any students out there or students to be who are listening to us? So can you tell us a bit more about how's your time divided between lab work and field work? How do you manage both? And can you tell us what do you do for each aspect?
So the division between lab work, field work, and sort of office stuff varies depending on a PhD. No two PhDs are alike. Even two people studying the same rocks aren't going to have the same sort of thing, the same kind of time divisions. So my field work was going to Australia to sample these rocks. And so that was very concentrated. I also go and do field trips with our undergrads and teaching and things. So I also like do that kind of field work. A lot of my time was spent in the labs. So the office work at the beginning was reading, and that got boring pretty quickly. As much as I like rocks, reading about them in papers can be quite dry. So as soon as I got samples back, I would take them into the lab. And for me, lab work was cutting up the rocks, polishing them, grinding them down, putting them on the microscopes, putting them on the electron microscopes. I had over 500 samples, and then I had to turn those into powders to go on different kinds of analytical machines. So I had to do all of that crushing, which was loud and noisy and smelly. And yeah, it took a long time, but I didn't really notice it at the time. It just kind of went by.
Yeah.
And then the rest of the time I was reading and writing or teaching.
Okay.
So
okay.
I was able to maintain a work-life balance. Like my supervisors didn't demand kind of undue hours off me, and I wouldn't have done that anyway because I worked before I did my degree and became a scientist. So I'm used to kind of doing 9 till 5 and being very kind of regimented in getting work done. So for me, it was very much like a day job. Started work at 8, finished at like 5 or 6, and didn't work the weekends.
Okay, great. So in terms of the lab work though, or in terms of the research, can you tell us a bit more about why is this research important and how it actually applies to the present or to the future?
So there's two main aspects, and one of them is kind of like, actually, I'll rephrase this. So when people ask about the benefits of science, and then what will happen is people will tell you about the features of a particular kind of science. So, but there's only really 4 benefits. A benefit either like saves you time and money, so saves you time, saves you or makes you money, changes how you see yourself, and changes how you see the world around you. So the benefits of my work is it changes how we see the world around us. We understand how the Earth evolved. How do you go from a barren planet to one covered in bacteria and then algae and then 2 carbon-based bags of water having this conversation right now, who've learned to take apart the universe and put it back together as laptops and tables and a Pokemon. And then change how I see myself is, I guess, ties into that because I kind of understand how the natural world works now, and I'm able to share that with, well, not how, that's a big thing. I don't understand how the natural world works. I understand it better than I did, and I can understand the impact that humans are having on the natural world. So I used to think maybe it's like, well, the climate changed its own quite a lot over geological time. How can I be sure that humans are causing this? Well, now I understand how ancient climates work and change. I can say, oh yeah, there's literally nothing else that can be causing this other than people. So that's another benefit of it. And then in terms of time and money, so these rocks hold resources, and we need those resources. The main one I'm concerned with are things like iron and phosphorus. Phosphorus we need to make fertilizer, and phosphorus reserves around the world are pretty low because life tends to take it and not let go. But if that life dies in a shallow sea, and then we find it billions of years later, we can maybe look at ways of extracting that phosphorus. Phosphorus also likes to soak up rare earth metals that we need for building things like laptops, telecommunications. So we need those. And then obviously we need iron, and iron deposits tend to be formed by the action of microbes or life concentrating that iron. So it all ties into how we understand like how the world works, how we're changing the world, and how life has interacted with the rocks and the atmosphere and such like to create deposits of the economic minerals that we need.
Okay.
Also, it's just really cool.
Yeah, I mean, you know, it's quite interesting. So, when you do your research, then are there any other research groups who might be collaborating with companies who might be helping to find these reserves for whatever reason?
Yeah, there's a lot of interest in the Northern Territories. So the other side of this is that these rocks are also full of oil, because wherever something dies, when you get a lot of living things die and buried, you get a lot of hydrocarbons generated. So these rocks are full of oil and gas, and the Australian government are interested in tapping into this reserve. I don't think that's going to happen anytime soon, because this basin, these reserves are all out in the desert. There's one road that goes through it. And you can only really get into it a couple of months a year, because in the dry season, it's too hot. Like literally nothing lives there. It's like there's not even kangaroos or anything. And then in the wet season, it's even hotter, but also it rains so much the desert turns to swamp.
Oh, okay.
So it's really difficult to get anything out there. So there's lots of groups working on it from the oil perspective, and then a few people who've been looking at the iron, where the rocks are close to the surface. But otherwise, it's actually, even though it's onshore, even though it's technically much easier to get to than, say, at the bottom of the sea where most, say, oil deposits are, it's proving very difficult for people to get out there. And also, there aren't that many geologists in Australia looking at it, so which is why we get to access to their stuff. And it covers a huge area of the Northern Territory. And so we're still mapping out where these rocks are under the ground.
Okay. All right, great. So I'm going to leave it there, though. And anyone who is interested to ask questions, please feel free to do this from the right-hand side of the YouTube channel, YouTube video. And then we're going to resume in about 13 minutes. So we will be back by 7:20. So I will see you all then. Thank you very much for your attention so far.
I'm just going to look for some photos now.
Hello. Thank you for joining back with us for the question and answer session. We're just waiting on Brooke, which I think will be here in a second.
Hello there. Sorry, I was talking to you and I didn't realize I'd turned my thing off.
That's fine. That's fine. Great. So, Brooke, would you like to start by showing us the pictures that or videos that you thought you had?
Yeah. Am I just okay to share screen?
Yeah, you should be a co-host by now.
Okay, excellent. Let's start with this one. So this is called the zebra stone.
Oh, nice.
It has all kinds of weird patterns in it. We don't really understand why. You only find it in one tiny valley in the Northern Territory, and it's not found anywhere else in the world.
Okay.
It's a complete mystery. So there's something very strange to start with. It's basically a mudstone that was deposited in a river. And that's mud. China clay is the beige bits, the yellowy, greeny beige bits. And then these red spots are iron oxide, but we don't know why it forms that pattern, because they have all kinds of different patterns. Some of them even look like writing.
Oh,
but
really?
It can't have been writing because they've been buried under the ground for hundreds of millions of years.
Yeah. That's so very interesting. Do you have one of those with what you think is writing, or which looks like writing?
I don't know. Not on this drive. It's on my computer at work.
That's fine. No worries.
Let's try another one. Okay, so here's one of the rocks. Oops. One of the rocks we found from the drill core. So if you imagine if this was vertical, the top would be this area here to the far right of the picture. And that's basically a beach, an ancient beach.
Okay.
And then all of these blobs and globules and wiggles and other weird shapes are little colonies of bacteria. And they basically secrete snot. We call it EPS if we're being fancy, which stands for extracellular polyramic substance. But I prefer snot because it's easier to say. And things get stuck to it. And
Okay.
the chemical, and within that slime, the bacteria can control the chemical conditions, and that allows certain minerals to grow really quickly, and then that sticks more things to it, and so they grow outwards as they get flipped around on the seabed and form these beautiful little structures. And this is an entirely new type of lithology that we've not seen before, because normally they're made out of calcium carbonate minerals, but these are made out of iron-rich clays, and no one's ever seen it like this kind of thing before. So, and also it just looks really cool.
Yeah, it does actually.
So
Very
I was saying about slicing them up and looking at them under the microscope. So one of those little globules under the microscope looks like this. Yeah, it looks like this.
Wow.
So this is under the polarizing microscope. So all the white grains are sand. So these are a mixture of quartz, feldspar, zircon, and other minerals that we call detritus because they get weathered out of rocks and just dropped everywhere. And this black gooey looking stuff is a mixture of fossilized bacteria, their poop, and the clay that they've produced. And if you look, there's a spiral. So this was probably on the seabed and then got ripped up by a storm and then rolled around. And so it's rolled up like a carpet. But the bacteria don't care. They just move up to the next layer so they can get to the sunlight.
Okay.
So if you want to imagine how small this is, if you look on the top left, there's a big grain of quartz.
Yeah.
That's about half the size of a grain of rice.
Okay.
So this is teeny tiny small.
Okay. That's quite interesting.
So we can actually zoom in even further as well. So here are some more. These are what we call ooids, and these are basically like bacterial bogies that have accumulated on the seabed and then got rolled around. They form in the modern day in places like the Caribbean, but they're made out of calcium carbonate there, and they're nice and white. That's what makes the white beaches in tropical locations. But here they're made out of all of this bacterial goo and these green iron clays, and they're green because there's no oxygen around. So even though this was on a beach, there was very little oxygen, so it couldn't get oxidized. So what we're going to do is we're going to zoom right in on these layers, because these layers accumulate. They're kind of like tree rings.
Mm-hmm.
So take that one away. So this one's about 2 millimeters across. So this one here, from top to bottom, is about the size of a grain of rice.
Okay.
But we can zoom right into it to look at those like the tree ring type structures. The laminate, we'll call them. It's just a fancy way of saying lots of layers. And look at them. So if you see those little wiggly bits,
Yeah.
you can see the dark bits between the layers that are look like made of little dots. Those are bacteria.
Mm-hmm.
So if you think that the big view was the size of a grain of rice, we've now zoomed so far in that we can see the bacteria that were living in it.
Okay.
So that's what we do with the regular light microscope.
Okay.
If we want to look close up at one of the bacteria, we use the electron microscope, and that produces something like this, a picture like this. So that's a 1.4 billion year old bacteria.
Okay.
So the outside of the polo would be the outside, the shell of the bacteria, the outside. And then the inside bit where it's hollow, that would be where the bacteria, the inside of the cell where its soup was. So I was saying that they were like a can of soup.
Yeah.
And then this is the phosphate, which it uses as food, the apatite.
Mm-hmm.
And it secreted it around its outside and then accidentally fossilized itself. So the modern ones do this. So if your oxygen level is going up and down really quickly, the bacteria have different ways of breathing. And to breathe when there's no oxygen around, they have to generate phosphorus, the apatite. And if it's without oxygen for too long, they basically fossilize themselves. And that's what happened here.
Right. Very interesting, though.
And then lastly, I said we had those slices of rock that we look at with the polarizing microscope. So I'll just quickly show you some of those. I'll start with something familiar. This one here, this spiral is a section through a snail shell. And this is from the Jurassic of North Yorkshire. So this is only about 200 million years old.
Okay.
And I'm just, there we go, rotating it round, and then I'll skip it forward a bit. That's a bit of shell. Oops. I'm just trying to get the bit where I switched to, so looking around and we can see them all. There's bits of all different sea creatures and shells that we're looking at that you wouldn't be able to see with your naked eye. These bits here, like that one that just moved, this is a bit of oyster.
Okay.
And all of this black stuff is sulfur.
All right.
And if you see, it changes color a little bit.
Yeah.
Okay, stop that one. And then finally, I'll show you one of the impressive ones. This is a rock called a gabbro, and this is what forms the crust of the ocean. This is what most of the Earth's surface is made of. This is what the deep seabed is made of. And in the middle, we've got a mineral called olivine, and this mineral comes from the Earth's mantle. It's come from hundreds, if not thousands of kilometers down. And then when the ocean plates are spread, it gets erupted onto the surface and forms the ocean bed. And when I said it changes color, this is, as it come up on the screen? Yeah, that's it changing color. So these patterns and colors, they're unique to olivine. So whenever we see this in a rock, we know we're looking at olivine.
Oh, okay.
And if you see these gray stripes, we know that's a feldspar mineral.
All right.
So this is how we can identify minerals.
Wow.
So if you see the one at the bottom with the pinstripes.
Yeah.
So I always like it when we show the undergrads how to do this for the first time, because they all sit there looking at it and it's transparent, and then they swap the polarizer and they get all the colors and the whole class just goes, oh, whoa.
Yeah,
So,
I know. It's quite
funny.
Nice, yeah.
If you looked at this rock to look at it in your hand, it would be a dull black, and you'd maybe be able to see some shiny crystal faces, but it'd just be black.
Oh, I see.
But by looking at it this way, we can see all of these textures, and this tells us about, this was originally liquid rock deep under the ground, and by looking at all of those different textures, we can see how the rock has, because we can't live in a magma chamber, so we can see how all of the rock has cooled and crystallized and what grew first and that kind of thing. We can just read it like it's a book. Yeah.
Great. Fantastic. Very interesting. Thanks very much for sharing, though. If that's okay with you, I would like to move on to questions from the audience. We have got a couple of these, but thanks a lot. Those rocks and what you have shown us, it's very interesting. So I would like to start one of the questions that I've received, though. I was wondering, can you tell us how long does it take you, from finding the sample to actually ending up with the images that you showed us? What is the time scale for that? How much effort?
So the longest part of it is shipping it back from Australia. So let's just say I went outside and picked up a rock. So around Oxford, there's lots of beautiful Middle Jurassic limestones that are full of fossils and cool things. So if I go outside and pick one of them up and bring it back to the lab, then I can have it under the microscope within a couple of hours. The longest step is waiting for the glue to dry when you stick it to the glass. That takes about an hour. But otherwise, it's just like you have to polish it you cut it up, and then you polish it on a polishing wheel, and then a finer polishing wheel, and then you polish it by hand with like a powder and water mixture into like a slurry. And then you have to do like this, and then check it, check it. And then you put it on a machine that measures it and cuts it down so it's as thin as one of your hairs. And you have to look at it and then measure it flat again, and then measure it flat again. And then you look at it. But yeah, a couple of hours.
I see. Okay. The other thing then I was wondering though, again, this is a question coming from one of our audience. So are there any ideas though why life wasn't diversifying during this era? Why did we only have microorganisms and not even like plants as we know it?
So there are a couple of main hypotheses, like ideas, why people think it is. Some people think that there was some kind of barrier in the way.
Mm-hmm.
Either there wasn't enough food,
Yeah.
there wasn't enough oxygen,
Yeah.
are the two big ones. And some people think that it just takes a really long time to evolve complexity.
Mm-hmm.
So if you think about it like this, life appeared on Earth almost instantly. As soon as you could have liquid water on the surface, life turns up. And then it took 2 billion years to go from bacteria to eukaryotes, the cells like us. But it only took 1 billion years, give or take, to go from single cell organisms, eukaryotes, to things like seaweed and probably jellyfish and sponges. And then 500 million years, more or less, to go from single-celled amoebas and seaweeds and sponges and jellyfish to us having this conversation. So every time it's actually got faster and faster and faster. And that's actually kind of a pattern that you see in other in other terms of like where you're getting complexity. So if you look at the level of complexity of, say, a civilization, it starts off slow and gets really fast. If you look at the development of computer technology, it starts off really slow and goes really fast. So the other argument says there wasn't enough oxygen and there wasn't enough food. So as well as oxygen for breathing, because oxygen respiration, basically using oxygen to burn food, that gives you the most energy. And one of your cells requires like 10,000 times more energy to power it than a bacteria. Bacteria can just sit there. There are bacteria that live in the rocks under the ocean, under kilometers of water and kilometers of rocks, and they'll basically breathe once every couple of 100 years. They don't need much energy, but what our cells do. And so if there's not enough phosphorus around and not enough oxygen, then we can't do that. Our cells can't do that, and that could hold it back. Also, oxygen does things like it allows you to build stuff like collagen. And if you've got collagen, which is kind of what makes your skin stretchy and lets you make muscles, then you can start going from a single cell to being something like a jellyfish with muscles that can swim and move around. So you need oxygen for that. Also, oxygen controls weathering on the surface. And that's where phosphorus comes from. So if you don't have oxygen to weather phosphorus out of the rocks, then things like cyanobacteria can't produce oxygen to weather phosphorus out of the rocks, and you get stuck in a loop. So what my research found, though, was that there was plenty of phosphorus. There was load. That's why they could fossilize themselves. There was too much of it. Not enough to poison them, but enough for them to be perfectly happy. Another idea says that there was lots of sulfide. Sulfide is really, really toxic to organisms like us because it gets in your respiratory system, and it has the same kind of shape as calcium, and it basically blocks up the calcium where calcium should sit in your breathing system and stops you from taking up oxygen. So a tiny bit of sulfide can make you ill pretty quickly. And so people thought there was a lot of sulfide in the ocean then, but then the work of myself and other people has shown that actually there wasn't that much sulfide. So we're kind of back to square one. And those of us who think evolution just takes a really long time are kind of going, well, and now we've also found modern organisms that would be perfectly happy in those environmental conditions. And so we're starting to think like, maybe they were, and we just don't have the fossils or the data, or we just think, that they look like bacteria. And the other thing is, evolution isn't something that has to happen. We tend to think of evolution because we get taught it in school, where you start off with like the single cell, and then you've got the guy with a beard at the end, because of Darwin, that things are trying to get to more complexity. Well, they don't have to be. That's why bacteria haven't really evolved. That's why something like an earthworm hasn't evolved beyond what it has. It's already pretty good at what it does. It doesn't have any pressure. And if there's no pressure there to evolve, then you're not going to evolve.
Okay.
But at the end of that billion years, there's a huge amount of environmental change, like gigantic ice ages where even the equator might have been frozen. And so all of a sudden, after a billion years of pretty steady conditions, you start to have all of these changes happening. So maybe that's got something to do with it. Question's still open.
Yeah, great. Thank you very much for this comprehensive answer, though. So going back to the climate. So so do you think, I've seen something here about the climate. So yeah. So do you think rocks or fossils can actually be used to indicate past climates or temperatures?
Yes. Yeah, and that's something we do routinely.
Mm-hmm.
So paleoclimatology, a lot of my colleagues do that. My supervisor, Stuart Robinson, and his group do that. My colleague, Ricky Sengupta, she was able to look at how much rain there was in southern England 90 million years ago through looking at rocks and fossils. So sedimentary rocks particularly, if they're well preserved, they record the situation, the environmental conditions at the time of deposition. And it's the same with creatures who make shells. They're recording the environmental conditions as they're making their shells. And so we can read that information, and we can combine that with laboratory experiments to kind of test how these chemical signals work, to test if they're actually realistic. And it takes a lot of jiggery pokery, but we've got it down to quite a good science now of being able to say, actually, we know what the temperatures were millions or billions of years ago. The other thing is, if you're at the South Pole and then you find a rock full of coral, we know that coral only live in tropical conditions. So either the South Pole used to be in the tropics, or it was tropical at the South Pole. So there's like a more practical way. Or if, for example, as some people have done, you find glacial sediments at the equator. Yeah. Were there glaciers on the equator, or were equatorial land masses that are now at the equator, were they at one of the poles? And then the answer to those questions is yes to all of them, because the continents move around, but the climate changes as well. Okay, all right, great, thank you. So how do you see then, continuing on the theme of climate, how do you think then, obviously we have seen a great change in our climate recently because of human activity. So how do you see that change impacts your field? You know, obviously fossils are forming as we are progressing through our lives. So how do you think the changes that we're seeing today is going to impact the geology in the future? So I'll start by saying that I'm not an expert on climate. I just know enough of it to be able to understand it from a geological point of view. So if there are any experts out there, feel free to correct me. In terms of, if I was to go into the future and sample rocks that are mud now, what we would see would be some very strange geochemistry. So for example, things like the oxygen and carbon isotopes stored in fossils would be very, very strange. We see this happen in the deep time. Say, for example, a giant volcano erupts and the Earth warms up, what we call a hypothermal event, which is the closest you can get to what we're doing. We see a big spike in the carbon and oxygen isotopes. So an isotope is where you have an element like carbon, and then you have a slightly different version of it. Kind of if you think of like carbon and diet carbon, or like carbon light, same with oxygen. And by measuring the ratio of the isotopes, the different types of the element, they relate to temperatures or to changes in like huge global cycles. So when we see ancient hypothermal events, usually caused by giant volcanic eruptions, when an ocean opens and a continent splits in half, we see these huge shifts in the carbon isotopes. And that's what we would see if you looked at sediments and shells from the modern day. There would be a lot of plastic as well in the rock record. There are already rocks now made of plastic from humans. So you would see lots of plastic and strange metal alloys and things in the rock record. Strange shell, like organisms might be incorporating things like heavy metals that we've released
Yeah.
For example, 'cause if you go back and look at the ice core records, you can actually see, for example, when the Roman Empire changed the type of coin that they're using, where they start including more zinc because it affects the global system.
Yeah. Great. Thank you very much. That's quite interesting. So again, there is another question from our audience. They're asking, what's the most exciting species that you have had opportunity to work on or to identify yourself?
Oh, good grief. Whichever one I'm holding in my hand at the time you asked me. Finding those bacteria and like realizing that I was looking and I was the first human to ever see them, that's pretty exciting. But I get that whenever I split open a bit of rock and I know that I'm the first human to see what I'm looking at, I'm like, oh my gosh, this is great. I got to go to a famous fossil site called the Burgess Shale, and I got to have a look at fossils and stuff there. And that's a place where soft tissue gets preserved, so you can see like the eyes and guts and things. And I found a trilobite, which looks like a woodlouse. It's like a fancy woodlouse, and normally it's just the shell, and they're really cool and beautiful. But this one, you can see its legs and its gills and its little feelers and stuff. And I was like, oh, this is so cool. But I get that excited. I got that excited yesterday. I was making a video about a fossil reef that's in Oxford, and I found a shell, and I was like, oh, this is so cool. So I don't think I could pick one.
All right. Okay. So the novelty is actually what is fascinating you about your research area.
Yeah, because you've got this connection to this thing. You're both alive under the same sky and the same sun, but just hundreds of millions of years apart.
Yeah.
It's the closest you're ever going to get to meeting an alien and visiting an alien world.
Yeah, I suppose you're right. Thank you very much. So it's very, again, very interesting. So I was wondering though, so I did have we have got quite a bit of questions from possibly prospective PhD students, I suppose. And they're asking about your personal experiences in terms of your PhD. So they're asking, what skills have you gained from your PhD, which are now helping you as a tutor? And where did you get your first teaching experience? And what is your favorite thing about helping us undergraduates?
Oh, good grief. Skills I've got from my PhD. So there's the obvious technical skills that I've got in terms of geology.
Yeah.
I've also got a lot of people skills. So I had a weird route into science because I worked for 10 years in call centers. I've done everything from call centers to crisp factories. And so I picked up a lot of what you call soft skills, like people skills there.
Yeah.
Before I came to do my PhD. So mostly what I've got from my PhD is the technical skills and thinking like a scientist. My first teaching experience is people, when I was on the beach looking for fossils, people would ask me about them, usually parents with kids. And so I would be explained to them about how to look for fossils, and I'm doing that. Like you can see, like the nodule I've just split open with the ammonite in it. And so that, but then when I was doing my undergraduate at Birkbeck, I would help if there were things where I'd picked up on something quicker, especially with field work. I would always help my classmates, especially like when they were struggling. So like helping your classmates when you're an undergraduate is a really good way to understand if you know it, because if you know something, you can teach it.
Yeah.
And then as soon as I got to Oxford, I was like, when can I start teaching undergrads? And what do I enjoy about teaching the undergrads? I get to talk about cool rocks with people that can't escape because they have to be in class. Seriously though, they're always asking like really good questions, and it's really fun when you do stuff like show them the thin sections for the first time, and they all go, whoa. That's really fun. And then helping them to understand the world and what's the point in me knowing all of this cool stuff if I'm just going to keep it in my head and like, you know, like the dragon with its gold. I always imagine people being like, who don't want to talk about their subject areas, where they've all just got it hidden away. Like it's there to be shared and utilized and to help people understand the world and themselves. So that's what I like about it.
Great. And another question again from, assuming from a prospective PhD student. So what do you intend to do after your PhD? Will you carry on teaching or researching or look for a job in a different sector?
So I've had normal jobs, and I really wasn't my jam. I'm not particularly interested in car insurance, so I'm probably not going to go back to that. I like rocks. I like teaching undergraduates, and I like researching. And the easiest way for me to do that is to continue on as in academia. So that's the current plan. Yeah, look for a postdoc and then go from there.
Lovely. Well, Brooke, I think this is the end of the questions that we had today. And I would like to thank you for your time. It was an absolute pleasure to have you. And it's really nice to see the examples of rocks that you have showed us. It's really interesting. And it's really nice that we actually get to hear from you as well, because normally people do assume that if you want to have a job in academia, you need to go through a certain path. And it's nice to show people that it's not, there isn't one way of doing it. There are multiple ways. As long as, you have the will, you will always find a way around it. So I'm sure it will be an inspiration for younger people.
Yeah, definitely. I failed my GCSEs and I don't have any A levels, but I still did a PhD at Oxford. So it took a bit longer, and I learned a lot of different useful stuff going through call centers and things, but it's definitely possible. It's like there's more than one way to get a job done. So
yeah, exactly.
If you want to do science or whatever as a career, you can definitely do it.
Yeah. Lovely. Well, thank you very much for your time, though.
You're welcome. Thanks for having me. I had so much fun.